Hi, my name is Asuka Eguchi. I am an assistant professor at the University of California, Irvine, in the Department of Physiology and Biophysics, and I will be sharing our work today on gene therapy for Duchenne muscular dystrophy. Duchenne muscular dystrophy is a severe muscle wasting disease. The skeletal muscles, diaphragm, and the heart are affected, and muscle degeneration is progressive. Patients typically rely on wheelchairs by the age of 12 to 14. And here you can see the muscle biopsy from a healthy individual and a Duchenne patient. And there is extensive scar tissue in the skeletal muscle, and there's a lot of variation in the fiber sizes. The prevalence of Duchenne muscular dystrophy is 1 in 5,000 boys. Current treatments include corticosteroids to reduce the inflammation, surgery to the spinal cord for scoliosis, physical therapy, and assisted ventilation because respiratory function is impacted. Patients typically pass away of cardiac or respiratory failure by their third or second or third decade of life. So there is this need to develop therapies for this neuromuscular disorder. Duchenne is caused by the lack of dystrophin expression, and dystrophin is an important cytoskeletal protein that transduces force from the contractile apparatus to the extracellular matrix. And this gene is encoded by the dystrophin gene, which is the largest in the genome. It is located on the X chromosome. And because it is an X-linked gene, Duchenne is a disease that affects boys, although female carriers can exhibit cardiac complications, particularly under stress, such as exercise. Without dystrophin expression, the plasma membrane becomes leaky, and this affects the regulation of calcium concentrations inside the cell. And since calcium signaling is really important for muscle contraction, this leakiness impairs proper and controlled muscle contraction. Dystrophin mutations come in all forms and they happen all along the gene. They can come in flavors of point mutations, large deletions, duplications, and inversions. And the most prevalent mutations are in exons 44 to 53, and then exons 1 through 21. Dystrophin, again, is the largest gene in the genome, so by sheer size, there is a higher probability of acquiring mutations. When patients exhibit delays in developmental milestones or exhibit issues with their muscles, they get tested for levels of creatine kinase, which are elevated in Duchenne patients. The average age of diagnosis is about 4 years of age. One of the classical signs of the disease is the Gowers maneuver, where a patient from lying on the floor in order to get to a standing position has to adopt this tripod formation and push against his thighs in order to stand up. And since Duchenne muscular dystrophy is caused by the lack of dystrophin expression, the most obvious therapeutic strategy would be to restore dystrophin expression by gene therapy. So gene therapy, we currently rely on adeno-associated virus or AAV, and this enables the systemic delivery of genes to all the tissues that are affected in Duchenne. And the promoter is the element that controls what tissues express the gene of interest, and the gene provides the coding sequence for the protein missing in patients. Currently, AAV is the best vehicle for gene delivery to all the skeletal muscles, diaphragm, and the heart, the tissues that are affected in Duchenne. The major challenge is that the packaging limit of AAV is 4,700 base pairs, and the coding sequence for dystrophin is over 11,000 base pairs. So dystrophin is 3 times larger than the packaging capacity of AAV. So decades of research in academic labs have focused on making the dystrophin gene smaller so it can fit into AAV. This case study was really the inspiration behind all the small variants of dystrophin. This individual naturally expressed a truncated variant of dystrophin with a major deletion, about half the size of dystrophin. despite missing this central rod domain. This person with Becker muscular dystrophy, a form of dystrophinopathy that is progressing more slowly than Duchenne. He was in the army in his 20s, climbed to the top of a cliff in his 40s, and walked with a cane in his 60s and survived into his 70s, following a very different trajectory of Duchenne patients who rely on wheelchairs by their teenage years and pass away in their 20s. His relative with the same deletion was a bodybuilder in his 20s, and his muscle biopsy is shown here. Compared to healthy individuals' muscle, you can see there is great variation in the fiber sizes. So indicative of this lack of dystrophin expression or this expression of this truncated dystrophin. And levels of dystrophin were measured by Western blot and using different antibodies detecting different uh, parts of dystrophin. And you can see that no expression of dystrophin is detected in the patient 1353 here, because this is the part that is deleted. Using an N-terminal antibody, this individual had very, very low levels of dystrophin compared to a healthy individual. And I want to emphasize that this protein or lysate was actually diluted 5 times in order to show this comparison. And then a C-terminal antibody also showed very, very low levels compared to the healthy individual. So they, despite expressing such low levels of dystrophin, this type of mutation caused a very mild form of muscular dystrophy. And this naturally occurring variant is still too large to package into AV. And so this gene had to be further miniaturized to fit into this vector. And studies by the Chamberlain lab, the Dwan lab, the Chow lab have demonstrated that these synthetic variants of dystrophin, about 1/3 the size of full-length dystrophin, can be packaged into AV and restore skeletal muscle function in dystrophin deficient mice. And so 4 of these miniature versions of dystrophin called microdystrophin have advanced into clinical trials. And the one by Sarepta Therapeutics called Elevidys was FDA approved in 2023. Pfizer ended their trial in 2024 because they did not meet their clinical endpoint by assessment of the North Star Ambulatory Assessment Score. And the clinical trials for Solid Biosciences and REGENXBIO are still ongoing. A major concern in terms of safety is that patients are being administered borderline toxic doses of AV gene therapy in order to achieve this level of expression depicted here. And this is a publication describing the Sarepta gene therapy. And patients receive between 1 to 3 times 10 to the 14 vector genomes per kilogram, which is an enormous viral load to receive all at once. And studies in non-human primates demonstrate that most of the AAV goes to the liver. And I'd like to point out that this y-axis is on a log scale. So compared to levels in the heart, the amount of AV that gets delivered to the heart is 100 times less, or 2 orders of magnitude less than in the liver. And while preclinical studies in mice have shown therapeutic efficacy in the skeletal muscle, we really do not know whether these smaller variants of dystrophin can prevent heart problems, because the animal models really do not recapitulate heart failure. And so mice that lack dystrophin expression follow a very different trajectory from humans, and they have heart problems at a very old age. They exhibit a normal lifespan and have mild muscle weakness. And by contrast, the lifespan of Duchenne patients is between 20 to 30 years of age. Also, a major limitation of the clinical trials is that uh, any patient that exhibits heart problems, either by echo or MRI, has been excluded from these studies. And it can take a decade or more for fibrosis in the heart or structural problems in the heart to develop. And so the study period of these clinical trials may be too short to see this clinical benefit with respect to the heart. But the heart is a really important aspect of disease because with advances in assisted ventilation, heart failure is now the leading cause of death, making it imperative that we address this problem. And Duchenne patients have a high resting heart rate even at the womb, and arrhythmia due to patchy scar tissue on the heart can be fatal. And disease progression to dilated cardiomyopathy occurs first with the heart cells or the cardiomyocytes that help the heart contract, and these cells die prematurely. The heart is not a regenerative organ, so the loss of cardiomyocytes leads to the infiltration of myofibroblasts and adipocytes that induce the replacement of muscle tissue with fibrotic scar tissue and adipose tissue. This causes structural remodeling of the heart, particularly the left ventricular wall. And this results in thinning of the ventricular wall, enlargement of the left ventricle, and then poor ejection fractions by echo. The current treatment is to slow the heart rate and lower the blood pressure using angiotensin receptor neprilysin inhibitors. This treatment reduces the mechanical load on the heart, but it doesn't address the root cause of disease, which is the premature loss of cardiomyocytes. To overcome some of the limitations of animal models, we use human-induced pluripotent stem cells to model the disease. By taking an accessible source of cells like the blood, we can reprogram the somatic cells to a pluripotent state, and these cells have the potential to become any cell type, including cardiomyocytes. And these cells spontaneously contract in a dish, and at day 10 of differentiation, they beat as a sheet of cells. And uh, shortly after the stage of differentiation, we dissociate the cells from each other and replate them so they form a monolayer of cells like so. And on day 30, we assess the cells for and compare healthy and diseased states. And on day 30, the cells are still spontaneously contracting, but each cell makes contact with the plate. And since each cell is adherent, you can still see the spontaneous contractions moving with a more subtle movement. And since these cells have the same genetic makeup as the patient, we can learn about the progression of disease in cardiomyocytes in the molecular context of human cells. By using multiple lines harboring different dystrophin mutations, we can perform clinical trials in a dish. Alongside each disease line, we have a healthy control line that has been CRISPR corrected, which means that the dystrophin mutation in these patients have been CRISPR corrected to create a healthy line that has the same genetic makeup as the patient, except for the dystrophin mutation. By using multiple lines and pairs of lines in the same genetic background, we can compare the effect of dystrophin deficiency and make robust conclusions about consistent differences between healthy and diseased states. So to compare gene therapy strategies, we differentiate the induced pluripotent stem cells to cardiomyocytes. On day 10 of differentiation, we treat the cells with either the gene therapy, microdystrophin, or an empty vector control akin to a placebo. And then on day 30 of differentiation, we assess the therapeutic benefit of various gene therapy strategies. There are some striking differences between healthy and diseased cells, and we use these readouts to assess therapeutic efficacy. The Duchenne cells are much smaller in cell size, and they express less proteins that help the heart cells contract. They also show poor calcium handling, showing these early after depolarization events and deviations from these 1 second intervals. And these cells are paced at 1 hertz, so you would expect the cells to contract and have these calcium transients at predictable 1 second intervals, but you see deviations in the disease cells. And the disease cells also beat with less force, and the Duchenne cells also show poor cell survival in a dish. So by using these different readouts, we can use these as a metric to measure disease outcomes and efficacy of gene therapy. So here we compared the 3 microdystrophins in clinical trials with the mini dystrophin discovered in the Becker patient who retained quite a bit of skeletal muscle function. The Duchenne cells exhibit poor cell survival. They also show a higher incidence of arrhythmia. And comparison of the gene therapies revealed that while the microdystrophins also have some modest impacts on the cell survival and arrhythmia, the mini dystrophin gene fares better. And this is the variant that was discovered in the Becker patient, and these disease phenotypes are rescued to a greater extent with the mini dystrophin. But this gene is too large to be packaged into AAV. And so a 2-vector approach has to be used for this larger variant. And we and others are delivering these larger variants, exceeding the packaging limit of AAV in a 2-vector system with split inteins. These split inteins enable these individual protein subunits to undergo transplicing in the cell, and then this approach enables larger proteins to ligate spontaneously in the cell and form these larger proteins. And we are also exploring delivering full-length dystrophin using lipid nanoparticles and encapsulating mRNA. In summary, the current gene therapies for Duchenne muscular dystrophy rely on adeno-associated virus to deliver small variants of dystrophin. Cardiomyocytes differentiated from human-induced pluripotent stem cells exhibit disease characteristics that enable these in vitro disease modeling to perform these clinical trials in a dish. And libraries of optimal gene therapy designs can be tested using this platform. With that, I would like to acknowledge my lab, my collaborators, and my funding sources. This concludes my presentation for gene therapy for Duchenne muscular dystrophy.